In this video, we will discuss about SRC deregulation and its impact on the cancer cells. So first we will discuss what is the difference between SRCC which is found in the normal cells and SRCC which is found in the cancerous cells, or the viral SRCC and circular SRCC. Though there are many differences, one of the main common difference that is presented in this figure is that there is a difference in the carboxyl terminal, that is tyrosine at 527 is present in the circular SRCC, whereas it is not present in the viral SRCC, and this will lead to the several interactions of this viral SRCC that cause its deregulations and can trigger several signaling pathways in the cancer cells. So some of those deregulations and how they impact on the cancer cells, we will discuss that thing. First we will understand how this viral SRCC will convert from inactive to active form. So there are different ways by which it converts from inactive to active form, that is protein tyrosine phosphatase, reduce CSK activity, displacement of intramolecular interactions, and these mutations that are mainly because of the deletion of the tyrosine at 527. So what happens in that case is that this kinase or this SRCC will interacts with the membrane through this binding partner, and this will lead to the activations of the kinase. That is, in the inactive form, the kinase region is interacted with this SH3 and SH2, but when it's SH3 and SH2 binds with this binding partner to the cell membrane, then it will lead to the activation of the kinase, and this kinase can act as receptors in order to trigger further intercellular signaling pathways. So here we can see that in the normal case, the phosphorylations at this 527 occurs, and that will leads to the attachment of the kinase to this SH2 kinase regions to the SH2, that is carboxylic with the help of carboxylic region. But in the case of this viral SRCC, this region is not there, so it's easier for this SRCC to comes from inactive form to active form. Now we will understand what are the signaling pathways it will affect. So here the combined signaling pathways of this SRCC and FAK when it interacts with the integrins is shown here. So what it will does is it will trigger several growth factors such as EGF and HGF, and these growth factors upregulations of the growth factors can further lead to this intercellular GF receptors that will cause activations of several signaling pathways such as growth factor induced mitogenesis or the C mice, and that we have discussed. This usually occurs because of the weakening of the membrane of the mitochondria, and this RAS, MEK, ERK pathways that will promote G1 and S phase, and the survival pathways, that is P13 kinase, PI3 kinase, AKT pathways, and the migration pathways will also take place through this P190 and Rho GAP that will lead to the change in the actins that are present in the cells, or the actin cytoskeletons get changed, and that will trigger the migrations properties in the cancer cells. So these are some of the pathways that get activated when this FAK SRCC interacts with the integrins, that will cause more growth factors formations that not only trigger changes the extracellular matrix but also triggers the intercellular signaling pathways. So this is the signaling pathways are because of the SRCC induced signaling from membrane that controls its cell behavior. As SRCC here we can see that interacts with the membrane through FAK. So I hope you understood some of the role of this SRCC, how it is deregulated, and what are the major changes that are found in the viral SRCC compared to the circular SRCC, and some of the signaling pathways that it triggered in the cancer cells. So that's all for this video. Thank you for your time.